Okay, as you well know, the CRISPR-Cas technique is a novel technique. It's being used with various types of living organisms, including plants. But due to time constraints, I will only refer to its use with humans. From time to time, the biological sciences undergo a technological breakthrough. Examples of this type of innovation in recent decades are the advent of recombinant DNA techniques, the cloning of the first mammal from adult cells, the establishment of the first cultures of stem cells derived from human blastocysts, and the generation of induced pluripotent stem cells from adult cells. We are now witnessing another breakthrough that seems to surpass the traditional recombinant DNA techniques in terms of versatility and accuracy, namely the so-called CRISPR-Cas technology. For those of you that are not familiar with the origin of this acronym, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. These correspond to DNA sequences in bacteria and archaea that, in conjunction with Cas proteins, Cas means CRISPR-associated proteins, participate in the recognition and in elimination of invading foreign DNA species, thus acting as an adaptive immune system. In particular, the CRISPR-Cas9 system offers a simple procedure for introducing various modifications at target loci in all kinds of living cells. This system relies on base complementarity between an engineered guide RNA and a specific DNA sequence in the genome, followed by double-strand cleavage by Cas9. The double-strand breaks are later repaired by one of several pathways that may lead to gene mutation or replacement. Recent developments have allowed the expansion of the genome editing repertoire of the CRISPR-Cas system. This includes modification of the guide RNA to improve recognition specificity, amendment of the Cas9 enzyme to create new docking sites in the genome, and deactivation of the nuclease activity of the Cas protein. A nuclease-deficient Cas enzyme can be fused to another enzyme to correct a mutant base or to introduce nonsense mutations at selected sites without cleaving the genome. Both the social media and the scientific community reacted very quickly upon the advent of the CRISPR-Cas technology. Their main apprehension is the possibility of editing the human germline either for therapeutic or enhancement purposes. The scientific community has taken several initiatives, consisting mainly in deliberations comprising both scientific and bioethical arguments, while at the same time encouraging further scientific research. A milestone in these actions was the meeting held in Napa, California in January of 2015, which is reminiscent of the Asilomar meeting that took place in 1975. Convened by the Innovative Genomics Institute, the group released a statement in which, among other recommendations, proposed not to conduct germline genome modification, at least for the moment. A similar conclusion was reached at the first International Summit of Human Gene Editing, convened in Washington, D.C. in December of 2015 by the U.S. National Academies of Sciences, Medicine, the Chinese Academy of Sciences, and the Royal Society. The second version of this meeting will take place in Hong Kong later this month. A voluntary moratorium as an effective way to discourage human germline modification has been supported by several scientists. For example, Edward Lanphier and 4 colleagues have argued that heritable human genetic modifications pose serious risks and the therapeutic benefits are tenuous. The NIH, for its part, has announced that although it will continue to support a wide range of innovations in biomedical research, it will not fund any use of gene editing technologies in human embryos. However, There are disagreements among scientists, as illustrated with these headlines taken from Nature magazine. Thus, Henry Miller from Stanford University claims that germline gene therapy should be used sparingly and with scrutiny, pushing the frontiers of medicine to rid families of monstrous genetic diseases. In a similar stance, geneticist George Church argues that banning human germline editing could put a damper on the best medical research, driving the practice underground. In a more cautious position, Jennifer Doudna, a prominent leader in the field, calls for urgent ethical discussions and avers that a complete ban is impractical given the ease of the use of the technique. In view of these disputes, it may be worthwhile to find out what science academies and societies have said in this respect. I have selected two of them based on their ample representativeness. According to the National Research Council in the US, the ethical norms and regulatory regimes developed for somatic cell therapy, this is for treatment of adults or infants, these rules are appropriate for the management of somatic genome editing implications. Now, in the case of germline editing for therapeutic purposes, research trials, according to the NRC, might be permitted, but only following much more research, and even then, only for compelling reasons and under strict oversight. In contrast, genome editing for enhancement should not proceed at this time, and public discussion should precede any decisions about such applications. On the other hand, the European Society of Human Reproduction and Embryology and the European Society of Human Genetics released a joint report asserting that although clinical germline gene editing would be totally premature, it might become a responsible intervention in the future, but only after adequate preclinical research conducted under societal oversight. It adds that the present prohibition in Europe of germline modification needs renewed discussion among relevant stakeholders. In the meantime, while these deliberations have been taking place, some research centers have dared to take the first steps. One would have thought that since somatic gene therapy offers much less bioethical apprehensions than germline gene editing, the first attempts would be directed to treat or prevent disease in infants and adults. Indeed, companies such as Editas Medicine, CRISPR Therapeutics, and Intellia Therapeutics, as well as research groups of various universities, are engaged in programs aimed at treating patients suffering from different diseases. To my knowledge, there are no publications in the scientific literature reporting actual trials. However, there is a short report note that appeared in Nature describing the treatment of a cancer patient in Chengdu, China, and a report in the Wall Street Journal earlier this year noting that there are presently in China 86 cancer or HIV patients being treated using the CRISPR-Cas technology. In contrast, scientific journals disclose that gene editing in human embryos is an active area of research, especially in China. To date, the experiments have been intended to test efficacy of the technique rather than to establish pregnancies by transferring the modified embryos. The first report appeared in 2015, implying that experimentation started as soon as the CRISPR-Cas technique was harnessed for gene editing in eukaryotic cells. Early results obtained in this work and a couple of subsequent attempts that you can see in the slide revealed several non-targeted gene modification events as well as mosaicism when the embryos were allowed to further develop in vitro. The protocols applied in subsequent instances were aimed primarily at overcoming these flaws. One of them involved the simultaneous injection into oocytes of sperm plus the editing components. This particular work, conducted in the US, resulted in a more efficient editing, although the interpretation of the results was openly debated by another group. In turn, a report by Huang's group from Guangzhou, China, deals with the amendment of the mutation responsible for thalassemia, a recessive disease that is caused by having two faulty copies of a gene. Because it would be difficult to find dozens of embryos that have this rare double mutation, the team developed embryonic clones from their patients' skin cells. There is also a publication by a British team in which, rather than targeting a disease, they investigate the function of the pluripotency transcription factor OCT4 during human embryogenesis. It's at the bottom in the slide. The CRISPR-Cas techniques exhibit some improvements, but it is still far from reaching a safety stage that would permit the transfer of the modified embryos or even its application in adult gene therapy. For example, it has recently been shown that cleavage of the DNA by the Cas nuclease induces the p53 mediated DNA damage response, leading to a selection against cells with a functional p53 pathway. The authors show that inhibiting DNA damage signaling improves the efficiency of editing in normal cells. However, inhibition of p53 leaves the cell transiently vulnerable to the introduction of chromosomal rearrangements and other tumorogenic mutations. In addition, work conducted with mouse and human cells has shown that CRISPR-Cas editing leads to large deletions and complex rearrangements both at target and distal sites. Still another setback to be solved, at least in protocols aimed at somatic gene therapy, is the identification in humans of pre-existing immunity to Cas9 proteins. It is for reasons like this that the New York Times felt compelled to publish an extensive article earlier this year cautioning about the present limitations of the CRISPR-Cas technology. Some final comments. It is likely that several features of the CRISPR-Cas are still unknown to us. Hence, it would be advisable for the moment to moderate the expectations and proceed with caution, especially when it comes to extrapolating the results obtained in the lab to the treatment of adult patients or human embryos. In particular, embryo editing constitutes a clear example of science advancing faster than ethical reflection and legislation, with the further complications that, due to different views regarding the moral status of the human embryo, general agreements are difficult to reach. More than ever before, the scientific community should be aware of its great responsibility in guiding society in safeguarding human dignity. Thank you very much for your attention.